All right, getting ready for Professor Pasquinelli's BIM 100 final? You are in exactly the right place. We're about to connect all the dots from lectures 5 through 10 and really turn these core concepts into a story that makes sense so you can walk into that exam feeling confident.
So here's our game plan. First, we'll hit the big picture, gene expressions control panel. Then, we're going to zoom in on turning genes on and off. After that, we'll look at how the cell perfects its RNA message. And finally, the most important part, we'll apply all of this to some actual exam-style questions. Let's do it.
Okay, first up, let's zoom way out. You know, we all learn the central dogma. DNA makes RNA makes protein. Simple, right? But the reality is way cooler and more complex. It's less of a straight line and more like a sophisticated control panel that a cell uses to manage its genetic information with incredible precision. And this right here is one of the most amazing facts in biology. Think about it. Your brain cells and your heart cells have the exact same DNA blueprint. So how on earth are they so different? Well, the answer is all about that control panel. It all comes down to gene regulation. Which switches are flipped on and which are flipped off in any given cell at any given time.
Okay, so let's get our hands dirty with that control panel. We're going to look at the actual switches and dials themselves, the molecules that are responsible for turning genes on and off by controlling transcription.
So a really big question is, how do scientists even know where these control proteins bind to the DNA? Well, one classic and really clever technique is called DNA footprinting. It's a way to find the exact landing pad on the DNA for a transcription factor. It's absolutely fundamental for understanding how any gene is controlled.
And here's how it works. It's pretty ingenious. You take 2 samples of your DNA, and one, you add the protein you think is binding. Then you add an enzyme that just randomly chops up DNA. Now here's the trick. Wherever your protein is sitting, it acts like a little shield, and the DNA can't get cut there. So when you look at the results on a gel, the control lane is a full ladder of DNA fragments, but the sample with the protein has a gap, a footprint, right where the protein was sitting. It literally shows you where it was.
Now, these proteins that bind to DNA, they generally fall into 2 main categories, activators and repressors. Activators are your on switches. They call in their friends, called coactivators, to help unwind the DNA, making the gene accessible for transcription. Repressors, well, they do the exact opposite. They recruit co-repressors to pack the DNA up really tightly, shutting the gene down completely.
All right, so let's say an activator does its job and a gene gets transcribed. The cell makes a pre-messenger RNA. But this first version is just a rough draft. It's not ready for prime time yet. It has to go through some really important editing and processing before it can be translated into a protein.
And there are 3 essential steps to this whole process. First, a special protective cap gets added to the front end, the 5' end. Second, a long tail of A bases, the poly A tail, gets added to the back. Both of these help protect the message and get it ready for its journey. And third, you have splicing, where the non-coding bits, called introns, are snipped out, and the important coding parts, the exons, are stitched together.
Now, splicing has to be incredibly precise. A mistake of even 1 letter could be a disaster. So the cell's splicing machinery, the spliceosome, looks for 4 key signals in the intron to know exactly where to cut. You've got the 5' splice site, a special adenosine called the branch point, something called a polypyrimidine tract, and the 3' splice site. And here's the key thing for the exam. If you get a mutation in any of those 4 signals, splicing is going to fail.
But wait, there's a problem. In our DNA, introns can be gigantic, thousands of letters long, while the exons can be super short. It's like trying to find a few small words in a giant rambling paragraph. So how does the cell manage? It uses a clever strategy called exon definition. Helper proteins bind to the exons themselves, basically putting little flags on them saying, hey, spliceosome, here's the important part. This makes it way easier to find the right spots to cut and paste. It's a beautiful system.
Okay, we've got the concepts down. Now let's switch gears and talk strategy. How does this stuff actually appear on the final? And how can you use what you know to nail those questions?
First things first, and this is straight from Professor Pasquinelli, you have to read every single word of the question and the answers. It is so easy for a question to swap one tiny word, like replication for transcription, and that one change can make a statement totally wrong. So slow down and be precise.
So you're almost guaranteed to see questions that ask you to interpret some experimental data.
For instance, you might get a question about a protein like the Huntington protein, HTT, and you'll have to figure out its function based on the results of an experiment they show you. This is a fantastic example. The key here is to only use the data they give you. You don't need to have memorized everything about the Huntington protein, you just need to look at the chart. If the bar for HTT with 30 repeats is really high, what does that mean? It means a lot of gene expression is happening. And what causes that? An activator. The answer is right there in the data.
Let's try another one. This is a very common exam scenario. Looking at some data and deciding if a drug is doing what you want it to. You might be asked to look at something like a chip experiment and say, is this drug a good candidate to turn on my gene?
And again, let the data be your guide. What are the telltale signs of gene activation? Well, we know that activators open up chromatin, and a key way they do that is through histone acetylation. So if the graph in the question shows that adding drug 2 causes a big increase in histone acetylation right at the gene's promoter, boom, that is your evidence. That makes it a good candidate. Don't overthink it, just follow the evidence.
And finally, there's another type of question where you just have to know your stuff. They love to ask what happens when you block a key enzyme with a drug or an inhibitor. This is a direct test of your knowledge of what these molecular machines do.
And a question like this is exactly why memorizing the functions of key enzymes is so important. If the question asks what an inhibitor is blocking, and the choices include RNase H and helicase, you just have to ask yourself, what do they do? You know that helicase unwinds DNA and RNase H removes RNA primers. If you know those basic facts, you can answer the question with confidence. It's a straight-up application of that core knowledge.
Okay, let's wrap this all up. Remember, gene expression is run by a complex control panel. Activators and repressors are the key players that open and close the chromatin. The first draft of an RNA message has to be refined with a cap, a tail, and precise splicing. And for the exam, the biggest thing is to read super carefully and always base your answers on the data they give you in the question. So that's our primer. As you keep studying, try to think about the big picture. We've talked about how one gene is controlled. But how does a cell coordinate thousands of these events all at once to build an eye or a brain or a whole person? It's pretty mind-blowing. You've got this, and good luck on the final.